Katira, who has been working very hard in cancer research, and he's gonna tell us a few things about his latest results, I hope. So
Yeah,
take it
this
away.
is okay. Thank you, Dr. Castillo. And it's always a pleasure talking at the CSRC. I don't have to worry too much about like what I'm gonna say. It's always, I don't know, it's like presenting to your family. So you get good feedback and it's nice. And let me see. Okay, so all right, can everybody see the presentation, the screen? And I wanted to test something else. Let's see. Yeah. Okay, so you can see the laser pointer. Yes. All right. So this is the third time probably I'm talking at CSRC in the last 5 years, and I've talked about different things, but I don't always remember exactly what I have talked about. So there might be a little bit of recap, but most of it is new stuff. Also, it's not something I have presented as many times, so it might not be as smooth, but we'll see how it goes. All right, so this is work mostly done by my graduate student, Benjamin Yeoman, who's a JDP student with UCSD Bioengineering, and it's in collaboration with Adam Engler's lab at UCSD. The focus is on mechanics of cancer. And why mechanics of cancer, especially when cancer is a biological disease rooted in DNA mutations? And the answer to that is because I actually know mechanics and I don't know biology as much. So mechanics of cancer works for me. But beyond that, also, I think there is something very specific about cancers and about tumors in terms of mechanical changes in the cells as well as the environment. So, for example, what happens when you have a tumor, right? For almost all solid tumors, so take lung, ovarian, kidney, pancreatic, breast, even brain, the cells, the tumor cells themselves are softer, they're squishier, and they're squishier compared to the normal cells. And the surrounding stroma is actually much stiffer. So the matrix actually is much more cross-linked, dense, and stiff. And this is true, right, across a lot of solid tumors, as I said. So then the question is, well, is this just a side effect, or does it actually play a role in tumor growth? And there are a lot of papers that have started investigating this whole mechanics aspect of cancer. One interesting question that comes up is, so imagine this, your cells are softer, the surrounding environment is stiffer. But cancer is actually a rapid proliferation and growth of these tumors, right? And so that itself raises an interesting question, that how are these softer cells actually better at pushing against a stiffer environment? It should have been the other way around, that, oh, you would have cancer if you had stiffer cells, which are now growing, and so they can push against a softer environment. But it seems it happens the other way around. So what's going on there? Then there is the other question, which has to do with migration, right? So the biggest issue with cancer is cancer metastasis, where the tumor cells will break out from the host tumor and then migrate through the surrounding environment, get into the vasculature, and then form secondary neoplasm somewhere else, right? So the cells actually have to migrate out. But for the most part, cells actually like to migrate to a stiffer environment, and we'll talk a lot about that. So then here what we see in cancer metastasis is actually that they're migrating from the stiffer environment out into the softer, healthy regions. So what's driving that? So that's another interesting mechanical paradox. And then the last one, it has to do with collective migration of cells. So cells, they have found that it's easier for cells that move as collectives. So not a single cell, but 3, 4, 5, 6, 7, or maybe 20, 30 cells together, when they move out and they go to a secondary site, they are more likely to form secondary neoplasms. Okay, so clusters of cells are better at forming secondary tumor sites, secondary metastases. But if you look at cancer cells, they actually don't like sticking to each other. And so then if you don't like sticking to each other, then how do you get along? How do you migrate together? How do you migrate as clusters? And so that's another interesting question. Now, I know for sure, so in the past, in this colloquium, in this platform, I've talked about the first question, where how softer cells actually are able to divide more because they are more mechanosensitive and they get more stretched out within the tumor environment. And so when that happens, they're able to proliferate faster. And based on that, we can actually show that if you have a small cluster of cells of softer cells within a stiffer, healthier tumor environment, they can actually start dividing at a much faster rate, now pressing or pushing against the surrounding healthier cells and sort of replacing them. And then we have also done some simulations based on what happens when you have soft and stiff cells in different ratios within a tumor environment, and what effect does that play on overall population dynamics, overall evolution of tumor, and stuff like that. Again, I'm not going to go into that. I've talked about this in the past, but if you have questions related to that, you can ask me later. The third question, which was about collective cell migration, and this is something we have done more recently, and we have simulations that look at what happens when you have clusters of cells migrating through a sort of three-dimensional fibrous matrix environment. So here you actually have these fibers all around. I'm not showing them because it would look crazy. And then you have clusters of cells migrating, and you have two possibilities. One where you have a single cell that's actually leading the whole cluster, and everybody else is following. And then you have a second possibility where each cell is kind of trying to do its own thing, where one cell sort of leads them in one direction, and then the second one leads them in the other direction, and then the third one can take over, and whoever is doing well will take over, and everybody will sort of follow this herd to some extent. And what we see is that depending on the kind of environment, different strategies, so either following a single leader versus this herd behavior, can actually optimize your migration behavior, and can also let weakly adherent cell clusters to actually migrate together, especially this herding effect. Again, not going to talk more about that. What I'm going to specifically focus on is the second question, which had to do with metastasis. And that was sort of the title of the talk, which was escaping the stiffness trap. And the reason I call this as a stiffness trap is that, so as I said, you have a tumor environment. The tissue or the matrix around the tumor directly is much stiffer. It's much more cross-linked. Okay, and as you go further away, it's sparser and less dense and less stiff. And for the tumor cell to metastasize, the cell has to kind of break up from this host tumor, sort of migrate through this environment and go through, and it has to basically navigate this stiff region and sort of get to the softer regions. Okay, and why is that a trap? It's a trap because if you actually, let me introduce a little bit about cell migration. So you take a cell, right, and the reason it's able to migrate is it's because it generates these forces driven by actin and myosin stress fibers. Okay, basically the cytoskeleton is made up largely of actin, myosin. There are a lot of other things. There are microtubules, there are intermediate fibers. They all play a role, but the role is secondary in regulating the actin myosin dynamics, and based on the actin myosin dynamics, that sort of triggers how much force the cell can actually apply. Okay, and then obviously if you're applying a force, what are you applying a force on? And you need to be able to apply a force on the surrounding environment. So you need to be able to grab onto the environment to actually transmit that force. And so that has to do with the adhesion strength between the cell and the environment. And then you have the surrounding environment itself. So think of this, right? If you are pulling on a soft spring versus pulling on a stiff spring, you're going to have a different effect. You're going to feel a different amount of force. You will experience a different amount of force acting on you, and so on. So that again drives what will happen. And combining all of these factors, what people have seen a lot is that almost all cells right? Almost all cells tend to migrate up a stiffness gradient. So basically, if I take a substrate or an environment where I have a soft region and a stiff region, and if I put cells in there, almost all cells, so almost all adherent cells, and not leukocytes or those ones, but your epithelial cells, your fibroblasts, so connective tissue cells, they will all bind to the environment and they will generate forces in a way that they migrate up this stiffness gradient. They like being in the stiffer regions. Okay. But just as we talked about, in cancer, the area surrounding the tumor, immediately surrounding the tumor, is stiffer, and the cell actually has to go towards the softer regions, which is what I mean as the stiffness trap. They have to sort of overcome the stiffness trap and migrate, which means that they need to basically somehow overcome this durotactic behavior. They have to be either adurotactic or even anti-durotactic, but they want to go towards sort of or against the stiffness gradient. They want to go towards the softer environment. So what could possibly drive that? Why would that be happening? And so what I'm going to talk about is related to, so this project actually started with our collaborators at UCSD, where they identified this mechanotype or this very specific niche of cells from a given cell population. And so this is not like, these are all the same same cells, right? So I'm not taking different types of cells or cells from different tumors or different patients or whatever. They're all the exact same cells, cell lines. So genetically, they're identical. And then you put them inside a chamber, let them stick to the slide, you flow your media through it, and based on the shear rate that you're flowing, that then applies a certain amount of stress on these cells. And so if you're applying a low amount of stress, shear stress, you look at what cells come out and you collect them. And so these cells are basically your weakly adherent cells, cells that are not binding that strongly to the substrate, because then they are able to lift off and taken out by this low flow. Then you put in high flow and collect everything else that comes out and you throw them away. And then you basically take the cells that are remaining, and these are now your strongly adherent cells. Okay, so you have sorted these cells based on how strongly they stick to your surface. Okay, so depending on how strongly they stick to the surface, you have sorted them into weakly adherent cells, which come off quickly, and strongly adherent cells, which are bound really strongly. These two cell types actually maintain the mechanotype over multiple generations and up to 14 days. Okay, and so this is not something which was just for the duration of the experiment that, oh, some cells came off or whatever. You actually take the weakly adherent cells and you let them grow again, and then you'll put them in there, and they will 80% of them will still come out at low shear. That means that 80% of them actually maintain that niche, right? And strongly adherent cells are actually even better in doing that. And so this is a niche that is preserved at least for a few generations and for a sufficient amount of time. Okay, so we have taken an isogenic cell line and separated them into strongly and weakly adherent cells. Interestingly, just this sorting based on adhesion strength gives you these populations which are different. So the weakly adherent cells actually migrate much faster compared to the strongly adherent cells, both in soft and stiff environments. And also, so again, not just in 2D, but in 3D environments, as you can see, the weakly adherent ones have penetrated through a three-dimensional matrix to much larger distances compared to the strongly adherent ones. And yeah, these are, again, how far they migrate and so on. Okay, so the weakly adherent cell type is much more migratory compared to the strongly adherent one. The weakly adherent phenotype is also more contractile. Yeah.
Sorry, one question. Can you go slide one, the pair? Yes. Okay. So here you're comparing the cell speeds. These WA and SA, what are the difference? Because you have stiff and soft, that's clear there. But
right, so the stiff and soft, yeah. So the stiff and soft is the environment.
Weak adherent and is that what?
Right, so stiff and soft is the environment, and WA is weakly adherent, right?
Okay.
And SA is strongly adherent.
And then you have that they're statistically significant. They're significantly different statistically.
Right.
Is that based on the mean? That the mean looks much more different than some of the other
So this is a
measurements
t-test.
there?
This is a t-test.
Yeah, but is it based on the mean or is it?
It's based on the mean and the standard deviation, right?
Mm-hmm.
I mean, it's
Okay.
on the entire sample.
Because they. Yeah, no. It's just that I wasn't seeing them as different, and I was surprised about being different. So I was just looking at those.
Yeah, they are actually statistically quite significant. I mean, it is based on the mean and the standard deviation, though, right? That's what the t-test does, right? Okay. Yeah, feel free to stop me whenever I'm not making sense. All right. So the other thing is the weakly adherent cells also generate stronger forces. All right. So this is something which is interesting. So they don't bind as well, but they're generating stronger forces. So this is very interesting because it's like, oh, you can migrate faster because you're not, as strongly stuck to the surface. Makes sense. But that's not it. It's not that they're not as well attached or they're not interacting as much with the surface. They're actually interacting, and they're interacting more strongly. They're generating much stronger forces. So this is basically this traction force calculated from this traction force microscopy. Basically, you have your substrate and you have these particles, and you track how much these particles displace as the cell is migrating on it. And then based on that displacement and the material properties of the gel, you can back calculate what the forces are. Okay, and so the weakly adherent cells are also more contracted. All right. Now, biochemically, what is different in quite a lot. So again, genetically, they are the same, but there is a difference in expression levels for over 500 different genes. We didn't go through all of them because it was crazy. But we do find that the difference is not actually coming from adhesion-related proteins. Okay, so the adhesion proteins are pretty much the same. They're as strong. But the big difference is actually coming from difference in cytoskeletal proteins. So myosin regulators, microtubule regulators, some GTPases, and so on. Okay, and so there are a lot of underlying. Obviously, it's not the effect is not just mechanical. It is driven by some expression level changes in the cell. All right. Okay. Also, this is an interesting thing. So we took the genotype of the weakly adherent cells and the strongly adherent cells and compared it to TCGA data, which is the Cancer Genome Atlas, which has patient survival data and their expression levels and so on. And what we find is that basically the gene expression profiles actually nicely sort patient outcomes. So the weakly adherent cells gene expression profiles that match weakly adherent cells actually show much poorer outcome in breast cancer patients. And it's relatively better for the strongly adherent phenotypes. Okay, so clearly this mechanotype actually relates to something that is seen clinically. Okay, also before we move on, so we have also done this, so we haven't done the gene expression analysis, but we have done the mechanical sorting and the difference in behavior for, so the data I've shown is for breast cancer cells, triple negative breast cancer cell line, but we have also done it for pancreatic and lung, and the mechanotype at least is there. We don't know about the genotype and how well it correlates to patient data yet, but the mechanotype exists in those two cell types as well. So it's not something very specific to just the cell line that we were studying. Okay, so then the question was, maybe this difference in the mechanotype also relates to the difference in the durotactic behavior, and maybe these cells, the weakly adherent cells, which are related to a poorer outcome, might be better at escaping the stiffness trap, or maybe more adurotactic. And so that was sort of the question. And to answer that, what my student Ben did was that he made these pattern gels. Basically, you have gels where you have regions of stiff and soft. And then you put the cells on there. And this is basically measuring the stiffness of the gels with AFM. And that tells you that you clearly have a difference in the stiffness. It's a pretty steep gradient, kind of similar to what we see in actual tumors, but the stiffness gradient is about 10 to 20 microns. Okay, and then you put the cells in there, and you can actually track the cells. Right? So if you are in the stiff region, you measure how far you are from the boundary, and yeah, you give it a distance. Basically, positive is on the stiff side. If you're on the other side, then it becomes negative, and you're on the soft side. And then you do that, and you can actually track where the cells are. So both those cells are in the stiff side, and you can do that for all the cells that you have. And what you see is this, basically, yeah, you can track these cells. And this is an automated tracking approach, again, that Ben came up with. And what you see is over time, you started off with the same distribution across, but over time, the strongly adherent cells ended up a lot more on the stiff side, while the weakly adherent cells didn't really care. They're still pretty evenly distributed. And so the weakly adherent cells clearly seem to be adurotactic. They do not care about the stiffness gradient. And keep in mind, this is not because they are not sticking well with the substrate, because they actually do generate stronger forces. And so they can interact with the substrate. It's just that they don't care about that gradient, or something else is happening. And we'll go into that a little bit deeper. Right? But so what we are showing here so far is that the weakly adherent cells are indeed adurotactic compared to strongly adherent cells. All right. And so what could possibly be going on? And so for that, we'll go back to sort of the early models of durotaxis. And the best and the simplest model of durotaxis, it comes from this two-spring model. So where you have a spring, which is your substrate, and then you have another spring, which is basically your cell-ECM adhesion. And then you have your actin-myosin, which is generating the forces that the cells require to migrate. Okay, so the cell forms a bond with the ECM. You're generating these forces. Now, since these forces are generated by these motors, which actually slow down based on the force that they are experiencing. So if you're trying to extract more force out of them, right, they will actually slow down. And so this is how they behave. So basically, how fast they move or how fast they can pull is dependent on the force that they are experiencing. So that means that if they start pulling, the spring starts getting sort of extended. Now they're experiencing more and more resistance, a higher force, and they'll start slowing down. Okay, and so as they slow down, basically you can sort of look at how the force evolves with time as this thing happens. All right, and that you can look at it as a function of K effective. Sorry, here the K effective is the same as this K, basically, which is the effective stiffness of the adhesion, which takes into account the stiffness of the substrate and the stiffness of the cell-ECM. Right, and what you see is that your force basically increases with time, and the stiffer higher your K effective is, the faster it reaches sort of this plateau. Okay, and so now think of this: if this bond has a finite lifetime, that the bond is going to break within a certain time, then your force is increasing at a certain rate. Okay, the faster your force increases, by the time this bond breaks, the higher your force. Okay, and so if that means that you're generating a stronger force or a higher force for a stiffer environment. And so now you're going, let's say you're at the boundary of stiff and soft, or you're going towards a stiffer environment, you're generating more forces before your bonds break on the stiff side, and so you're getting pulled more towards the stiff side. And that's sort of the basic model for durotaxis. And I said, like, almost everybody has been focusing on durotaxis because almost all cells durotax. Even the population that we showed, if we just throw in the raw population as is, we'll see a lot of the cells durotaxing. But what we now separating them based on this adhesion strand, on this mechanotype, you can actually see that there is a distinct type of mechanotype of cells that actually adurotax if they don't care. All right, so yeah, adurotaxis is something uncommon. People have been working towards explaining durotaxis. So this is the early initial model, simple, beautiful, works nice. Obviously, it's too simple for biologists, and they added more complexity. And so then you also have some feedback mechanisms where depending on the force that the myosins are applying, you have more and more myosins which are getting activated and generating forces. And so in general, your overall force that you also apply can increase. And so that's another model for durotaxis. There is a third model for durotaxis, which looks at basically kind of this effective stiffening of the spring as you go closer and closer to stiffer regions. Basically, think of this: you have your fiber or your matrix environment, which is a connection of springs. You are pulling on it. The more springs you have, the weaker that effective spring stiffness is compared to like a really stiff rigid environment. And the closer you get to this rigid environment, you'll feel that stiffness more and more, right? And so that keeps driving you closer to that stiffer environment through the same mechanism of increased forces on stiffer environments. And so again, more explanation for the durotactic behavior. So far, nothing about adurotaxis, and this is all the way to 2017, and even 2019, you have more and more papers on explaining durotaxis, right? More complicated models, different approaches, continuum, discrete, so on, but nothing talking about adurotaxis. Finally, we found a paper that actually does talk about adurotaxis, and this was pretty cool because, okay, you have what they do is based on, again, similarly based on a niche of what the cells are doing, right? How much force the cells are applying, they can actually explain durotaxis and adurotaxis. And what they take into account, which is very important, which the other models kind of ignore, especially this first one here, right, is that this bond lifetime is actually not fixed. So depending on the force that the bond is experiencing, the bond is actually going to break faster, right? If you know the Arrhenius equation, basically you're lowering the activation energy to sort of break the bond, and by applying this force, and so you would break the bond faster. And so now your lifetime of the bond is actually also decreasing as your force increases, and so you sort of have an optimum, and that's what these people find. So basically they took into account the effect of those forces on the bond lifetime, and so you have the motors generating these forces on your actin filaments. Actin filaments are bound to these clutches, as they call it, which are Cell-ECM adhesions, and then you have the substrate stiffness, and you're generating the forces, and the bonds will break and form and break and form depending on the strength of the forces that they're experiencing. And what they find is that for every cell, you sort of have this ideal stiffness region, okay? Optimal stiffness where they are the most migratory and they want to be there and they are happy, right? But the problem with this, so we should be like, oh, so this explains adurotaxis, because if you're in a region which is stiffer than your optimal stiffness, you would prefer to go back to that side, right? And that could explain adurotaxis. The problem with that is that as you increase the contractility of the cells, as you increase the force that the cells are applying on the substrate, right, that region actually migrates towards the stiffer side. The optimal region actually migrates or shifts towards the stiffer side. And so more contractile cells should be happier on the stiff side, not on the soft side. Sadly, what we see from our experiments was that the weakly adherent cells, which are more contractile, prefer or particularly don't have a preference on the soft or the stiff. And it's the weakly contractile ones or the strongly adherent ones, but generating less forces, they like the stiff side more. And so this model exactly doesn't quite fit what we are looking for. And so we went a little bit deeper into this whole effect of bond dynamics and forces. And the idea here is that durotaxis happens when you have a higher force on the stiff side, lower force on the soft side, or a higher bond lifetime on the stiff side, or a lower bond lifetime on the soft side. Basically, you are sending out these protrusions, right? And you're generating a force. If you're generating a stronger force on one side, then that's the side you're going to go towards, clearly. But also, if you're binding to that side more often, right, then probably, yeah, it's a balance of how much force you apply and for how long you're applying the same force, right? So that balance gives you durotaxis versus adurotaxis. And so maybe you're generating, you could also have, it's ideally a balance, right? So you could generate lower forces, but if you have a higher bond lifetime, then you could still go in the other direction, and so on. And so looking more into that lifetime problem beyond just the force, because force everybody has looked So to go into that detail, this is a quick idea of the complexity of the cell-ECM adhesion. So you have the actin fibers, you have a bunch of adhesion proteins that bind to your integrins. The integrins actually bind to your ECM. You have a lot of other proteins that actually bind to this integrin actin complex and sort of generate secondary signals that will add more integrin filaments to it, that will actually also allow clustering of integrins together and so on. So it's a very complicated thing. We have tried capturing most of this within our model, but the key aspect here was that the integrin ECM bond themselves are actually not your standard bonds, which are slip bonds. So slip bond is basically where you apply a force and the bond lifetime decreases. What you have here instead is a catch bond, which is basically where you apply a force. So this is done using an AFM, and you pull on the integrin ECM bond, and for a while your bond lifetime decreases, but then your bond actually starts getting stronger as you apply this force. And then once you go beyond a certain threshold for the force, then the bond again weakens. And so the integrin ECM bond is not your regular slip bond. It's actually a catch bond which strengthens on the application of a larger force. And so that actually is pretty interesting again, right? So think of this, you have your catch bond versus your slip bond. So you have your strongly adherent cells, okay? And on a soft substrate, they're generating a lower force, as we talked about. On a stiff substrate, they're generating a slightly higher force just because as your stiffness increases, your forcing application increases. All right. But then your bond lifetime is lower on the soft side, higher on the stiff side, and you could see that why these cells would sort of migrate towards that. But then you could also have a different niche where you have these weakly adherent cells which are generating stronger forces. So on the stiff substrates, they're generating a much stronger force compared to what on the soft substrate, but they're still generating larger forces compared to the strongly adherent ones. So they have more contractility, they're generating larger traction forces, but the bond lifetime has now gone down actually compared to the soft on the stiff side. And so now you have this imbalance in the bond adhesion lifetimes that you want across your barrier. And using that, you can actually sort of identify this force regimes where you would have this imbalance between force generation and bond lifetime, right? Yes, Anka.
So for a non-expert,
right.
What's the molecular basis or physical basis behind the catch bond thing?
Well, that's an interesting.
How does that work?
So, right, and the way it works is actually it is think of your normal bond. Basically, what you show it is something like this. Okay, so you have a pocket and you have a binding site. It sort of fits in, and you generate a force, and you're trying to pull it out of the binding site. But now, if you have a binding site that looks something like this, where you have a pocket and the bond actually starts off here, and you're generating a force, so it could actually pop off. But if you generate a strong enough force, it could actually go in deeper and form a stronger bond, right? And it sort of gets entangled. So yeah, you're trying to sort of form this bond, and then you kind of reach this region. It's basically your binding site architecture is slightly different. Again, now the molecular biophysicists, I know there are quite a few in this. You can see Tom right there. But yeah, they can probably explain more on how it works, but this is how I understand it, at least. But yeah, so that's your sort of a catch bond, which strengthens on generating a force. And so what that could do is it gives you sort of this force regime where you could, when you are on a soft substrate and you're applying a certain amount of force, you're basically at that optimal force for your catch bond, and so you're strengthening these bonds, and so you can generate, you're basically not breaking as often. And so that gives you these, the blue lines, which are basically your strongly adherent cells with lower traction forces or lower contractility, which have a higher lifetime. And so that means that they're forming stronger bonds on stiffer substrates, which actually kind of correlates to or tells us why they would be also strongly adherent, not that easy to break off, right? Compared to these more weakly adherent cells, which are generating stronger traction forces, they have more actin-myosin force or contractility. And so their bonds are actually weaker as you get onto the stiffer substrates. And so they kind of come off faster, right? And so you can sort of create this niche just by tuning the contractility or the actin-myosin force that your cells are applying on the substrate, which sort of gives you this niche where you're generating stronger traction forces as you increase your substrate stiffness with the stronger forces, but you are weakly adherent on higher stiffnesses, right? And so now we take this niche and we build a model where you have your cell center, you have the stress fibers, basically your actin-myosin, which can polymerize and depolymerize. You have your binding sites to the substrate. So these polymerizing actin-myosins will actually find these binding sites, stick to them, start generating forces. You can actually, we also have the strengthening of these sites by fiber reinforcement. So if you have these stress sensors, which are proteins such as paxillin, then the stretching of paxillin allows additional stress fibers to come in and bind. And so you could actually have more forces that are being generated at the same spot. You can also have branching within these stress fibers. And so you take into account all of these dynamics along with the binding and unbinding based on the catch bond and the max force that you can generate within the actin-myosin stress fiber, sort of within this range of 30, from say 10 to 30 piconewtons, which would be a strongly adherent phenotype, and then higher than 30, somewhere 30 to 50 piconewtons or 30 to 60 piconewtons will be your weakly adherent phenotype, right? So the only thing we are changing is actually this max force Fs that can be generated in the stress fibers. Everything else is the same. And we run the simulation, and you have a cell which is sort of generating a bunch of these and migrating around. You can actually put them right at the interface, and you can see how much force is being generated on one side versus the other. And what you can see is that for the green one, which is basically your catch bond, right? So the red one is your slip bond, and green is, your red line is basically your forces along this direction. Your green are basically forces in the direction of the gradient or away from that, right? And so you have a catch bond, and you're changing the maximum stress fiber force. And what you see is that up to a certain value, you're increasing the force in the positive X direction, which is towards your stiff side. But then beyond that, so if you have your cells which are generating much stronger forces, then as your force actually starts going down, or is generating lower forces, or smaller forces, or forces less directed towards your X direction. So they're evenly distributed on either side. So the positive value is towards the positive X, the negative value is towards the negative X. And these are averaged out. So basically this tells you that up to a certain point, you will see this durotactic behavior. Beyond that, you'll actually see more adurotactic, right? Okay, and so you take that, you simulate your stuff. This is your simulation on stiff and soft pattern gels, and you see the behavior that you see experimentally. So your weakly adherent cells, which are basically generating lower actin-myosin forces, are more durotactic, less adurotactic. Your weakly adherent cells, which are generating stronger actin-myosin forces, are less durotactic, more adurotactic. Again, you can also see kind of exactly the same redistribution behavior for these simulated cells. Okay, all of this is good, right? The only thing we are differentiating our cells is based on how much actin-myosin force that they can generate. And based on that, we are saying we are regenerating the mechanotype, which is more contractile, less adhesive, and it shows you the differences in durotaxis. The question is, can we actually test this out experimentally? All right. And so what we're saying is that depending on your actin-myosin force, right? If you start off with a strong actin-myosin force, then you are adurotactic and you don't care. But as you lower that the actin-myosin force, you should start shifting towards more and more durotactic behavior. Okay. And one way to do that in the experiments is actually by reducing myosin activity using the small molecule called Metastatic. which interferes with myosin 2 activity, which is myosin 2 is what generates the forces in most of these cells. And so if you interfere with this myosin 2 activity, you should be able to see that exact same theoretical prediction. And so here we actually tried doing that. And first of all, what we see is that if you take your weakly adherent cells, so this is just for weakly adherent cells, and you treat it with blebbistatin, they actually slow down in speed, right? Similar to your strongly adherent cells on both the soft and stiff substrates. Okay, so that's good. And actually, so this is your weakly adherent phenotype. This is the experiment. As you put in more and more blebbistatin, you're seeing more and more accumulation towards the stiffer side. You're actually seeing these weakly adherent cells now becoming more and more durotactic. So clearly, by just changing the myosin contractility, you're not doing anything else further down into the biology of the cell. You're just tuning the mechanotype. All right, and so that's where I will conclude this talk. I have no idea how long it took. Hopefully, it wasn't. Yeah, okay, not bad. 40 minutes. Basically, your durotactic behavior that we see, or durotactic versus adurotactic behavior that we see, is highly dependent on cell contractility. The mechanotype, right, that comes out of this contractility, which is a weakly adherent mechanotype, contributes to a higher metastatic potential and lets the cells escape the sort of the stiffness trap that I talked about. And finally, we have a mechanical knob. So we're not, again, going and treating anything else that is wrong with the cells. We're just tuning the contractility, right? And by tuning the contractility, we can actually convert these weakly adherent cell types to behave more like the strongly adherent ones and become more durotactic and hopefully less metastatic. With that, I'll obviously Ben did most of this work, and then we collaborated with Adam and his PhD student Pranjali, just graduated. And then that's the rest of my lab. Esteban especially contributed to sort of the actin myosin force determination and those models and so on, and then our funding sources. So thank you for listening. And I see 2 hands already raised. So I'm happy to take questions. All right, so Anka, I guess.
Oh, sorry, I forgot to take my hand down.
Oh, okay. So Sandy?
Hey, nice talk.
Thank you.
I wanted to ask, of course, a biological question. Are these cells dividing? Cells that divide tend to be less adherent, and they're also smaller in size, at least at the early stage.
Yes, absolutely. So the cells can divide. We're not blocking adhesion. We don't take into account the dividing cells. Again, the observations are within 24 hours, so we don't have too many of them dividing within that time frame. The one thing is that we don't see a difference in their proliferation rates or division rates between weakly adherent and strongly adherent. So they're dividing at the same rate. So it's not like one is dividing more than the other. The other part that you said, like if the cell is undergoing division, and they are weakly adherent, and that's absolutely true. So when we're trying to sort the cells out, right, we might actually get some of these cells which are not necessarily weakly adherent, but just at that stage of mitosis or entering sort of the M phase, where they are now rounding up and they're less adherent, and so we're actually capturing them. And that could possibly explain that why we don't see our weakly adherent population when we just grow them, say completely weakly adherent, but we see some of them actually become more strongly adherent within the first to the second generation itself. And so that might be just because we are capturing some strongly adherent cells, which might just be dividing at that time. But yeah, I don't know what the right way to correct that.
Yeah, I mean, the weakly adhering ones, you could make an argument they're just more metastatic if they're dividing more rapidly and tend to, to move.
Right, so but the proliferation rates are almost identical for both those cell types.
Okay.
You separate them out and then you culture them, and then you look at the proliferation rates and they grow at the same rate.
Thanks.
So, sure. I think Carrie.
Hey, great talk, Parag. My question is kind of like so I think I missed what cell line you used. Was this a breast cancer
So this is 231. Yeah, this is MDMB 231. So this is triple negative breast cancer. But we have also done it with a lung cancer cell line, HT something, I forget, and a pancreatic cancer cell line too.
And this is, so the model is consistent like across tumor types?
Yep.
Okay. That's cool. And
yeah.
Have you ever done any like single cell sequencing between like the
right yes, so we did RNA-seq basically on that, and yeah, there are
of like the bulk population or single cell sequencing?
No, this is bulk, but sorted bulk population, right? So sorted weakly adherent bulk population and sorted strongly adherent population.
Okay. Yeah, I was just a little curious, like what the heterogeneity is like within the weakly adherent, the strongly adherent populations. Because like you were talking with Sandy, like yes, maybe the more proliferative cells are metastatic, but sometimes actually the metastatic cells are not very proliferative, or they go through these phases where they're not proliferative while they're metastasizing, et cetera. So there's kind of a lot going on during that process.
Right. I think that is the question that I've always had, which is basically migration versus proliferation. And cells that are more migratory, I'm guessing, would tend to be less proliferative. Or rather, if you slow down proliferation, then they tend to be more migratory because they want to get to an environment where they can proliferate more. But that is just my guess. I've recently seen a paper where they show that that is not necessarily true. They have migratory cells which are as proliferative as non-migratory cells.
Yeah.
But at the same time, I've also seen papers where you either induce hypoxia or some other sort of stress signal which sort of prevents the cells from, or tells the cells don't divide. They all of a sudden become way more migratory.
Yeah, it's like allocating resources to do the things they need to do. But I'm sure there's obviously some variability in that population that's actually strongly adherent or weakly adherent and moving.
Right.
Okay. Thanks.
Yeah. Sure.
Tony.
Yeah. Tony.
Parag, great talk. A basic question, and can you explain again the rationale? I got a little bit confused on the weakly adherent cells.
Right.
Are less or more contractile, and how that relates to the critical force that you were actually modulating to generate the phenotype.
Right. So the weakly adherent cells are more contractile. They're generating stronger traction forces. Okay. And that's the experimental observation. And that's sort of what kind of drove us to, in our model, to increase basically the rapamycin force. Right. So now think about this. Where that comes in is this value Fs. This Fs is the maximum force that your actin-myosin system can generate, right? It can sustain. Basically, your actin-myosin stops pulling at that force, okay? And that is dependent on the number of motors that you have. Number of active myosin heads you have will change that stall force. Basically, the more number of myosin heads you have, you can sustain larger forces, okay? And so that's your Fs, which is basically the max force that you can generate in your filament. And that sort of gives you this, right? So if you think of this, you have a cell with a higher Fs, sure, it's going to generate a greater force, right? Period, on a higher stiffness. But the question is, how does that affect your adhesion lifetime? Because now you're generating higher forces that might actually weaken your bonds. And so that's what happens when you have these catch bonds. Now, where you are actually generating higher forces, right? But your bond lifetime is lower on the stiff side. And even though on the soft side, you're generating lower forces, your bond lifetime is higher because you have, so this is your, say, strongly adherent cell. This is your weakly adherent cell. So your strongly adherent cell is generating on stiffer substrates, you have a higher force, but it's not high enough to affect the bond lifetime. Right? And then on the soft side, you have a lower force and lower lifetime. And when you generate even larger forces for the weakly adherent, then you are actually generating higher traction forces on the strongly adherent, on the stiff side, but your lifetime has gone down significantly, where your lifetime is higher here. And that sort of balances this out, where you no longer see this durotactic behavior because, hey, you integrate your loading, ideally, right? Force versus time. And you end up sort of balancing the whole thing out. Does that make sense?
Yeah, that makes a lot of sense. And the other question is following up on what Anka asked before, is could you explain on this, or your interpretation? It seems that this catch bond is key to understand the variability of your results on the different stiffness of the tissue that you're working on.
Right.
But can you explain on the context of your model or your cells from the mechanosensing point of view? What's going on, or what do you think that it's going on that is creating this catch bond? Because before you made an illustration assuming that there is some topological, for example, complexity that might lead to this, but how do you see that in this specific system?
So not exactly, well, again, right, so the previous models, they don't actually take into account bond dynamics. And if they do take into account bond dynamics, they are generally focused on slip bonds. Okay, and so that means that the stronger the force you're applying, the more quickly your bond is going to break. And so what that does, I don't know if I have that plot in here. Let me see. Right, and so you can look at this, right? So here, the dashed lines are basically what you would get if you had slip bonds. And so what that tells you is if you had a slip bond, again, your substrate stiffness increases, your traction force increases, which is fine, which is with the previous model, right? Your substrate stiffness increases, your traction force increases, and higher your actin-myosin stall force, the higher that contractile force is. That's fine. As your substrate stiffness increases, your bond stiffness or your bond lifetime goes down. Okay, and you're going to break the bonds faster. But for most relevant stiffnesses, so this is sort of where your healthy mammary tissue is. This is where your sort of your tumor tissue is, right? In the case of the slip bonds, there is no difference in the lifetimes, in the average lifetimes. You're already sort of way past that breaking rate where you have sort of maximized your breaking rate. You're just breaking quite fast. And so then it's clearly driven by the differences in your traction forces. And so if you just had slip bonds, then you will always see durotaxis. So if you look here, right, right, your slip bonds, you will always see durotaxis because, yeah, you keep increasing your max force or whatever, and yeah, you're generating larger forces, no difference in bond lifetimes. But now if you go to catch bonds, as you can see here, you have a higher force that you're generating, right, with stiffness for both of these weakly adherent and strongly adherent, or rather weakly adherent and strongly adherent. But your lifetimes are different. So your strongly adherent one, your lifetime actually goes up because of the catch bond, because you're now at that optimum strength of that catch bond and you're actually stable. But your weakly adherent one, which is generating larger forces, sort of pushes you over that hump. And now your bond is breaking faster. And so your bond lifetimes are lower. And sort of that balances out. And so the catch bond dynamic seems to be critical in sort of explaining this adurotactic behavior by strongly or by more contractile cells of weakly adherent cells. That
okay, thanks.
Sure. Other questions?
I guess
I can say something about catch bonds maybe with myosin actin.
Right.
The myosin and actin interact and have ADPs stuck in the pocket, and it's thought that the catch bond actually keeps the ADP in there, preventing the dissociation, you know, just by the physical movement of the myosin relative to the actin. And that dissociation of the ADP is necessary for the release eventually when of the ADP when the ATP comes in. So in other words, it strengthens the bond by keeping that ligand stuck in the pocket as a result of the physical movement.
Yeah.
I don't know if that's helpful or not.
No, that's absolutely, yeah. I mean, it's basically protecting the release of ADP as you stress it more.
Right.
And yeah. And yeah, you were there in Carly's thesis where we actually had the catch bond dynamics in the actin-myosin bonds as well.
Right.
So yeah. We have some more stuff on that. Esteban is actually working on that stuff now. So, but at some point, yeah, we'll talk to you about it.
Any other questions?
Or comments?
Comments, complaints,
Yeah.
compliments?
If I made you fall asleep, whatever.
Maybe that's why they're not asking. So well, and that is the case. So I would like to thank you, Parag. It was a wonderful, enjoyable talk, as usual. And wish everybody a happy Friday afternoon. And we owe you the trip to Eureka, hopefully.
Yeah, hopefully soon.
Yeah.
I need one.
Okay.
All right.
Bye, everybody.
Thanks, guys. Bye.